Tim also probably from her prior roles as well, no doubt, but I'll leave it to you, Lynn, to lead us on that.
This is odd, I'm expecting Martin to walk in any second. Someone tell me what room it was in?
Yeah, we did. We've put a couple of feelers out there, so we'll...
Okay, right. Well, let's start. I'm not Martin, I'm Lynn, and I'm going to talk a little bit about the sort of research that I've been involved in, but as I mentioned last night, it's not me, it's a team of people. So we set up something called the Genomics Research Centre a number of years ago, about 10 years ago, and we brought that name with us from our whole lab moved across from Griffiths University to QUT about 6 months ago. I think just about everybody from the lab moved, which means that a number of people sort of drive up and down the coast a little bit for those who still live there. And they don't seem to be too mad at me. But we had the title Genomics Research Centre well known for a number of reasons, and so we decided to bring that with us, and we have permission to do that. So that's now actually been transferred across to QUT. So this particular centre undertakes research to try and identify genes involved in common human genetic disorders. We do that so that we can look at how those genes interact with each other, so that we can work out whether there are environmental and genetic components, and so that we can work out what's going wrong in terms of perhaps gene structure or gene function in relation to disease. We also translate that work by undertaking diagnostics and therapeutics. So our aim is to develop better ways to treat people, but also better ways to diagnose people. We work on a number of common, what we call genetic disorders, but maybe you don't think of them as genetic disorders. And these are really common disorders like migraine, like multiple sclerosis, cardiovascular disease, osteoporosis, glaucoma, and also various types of cancer. So we work on a number of disorders. It's not focused on just one, but I suppose the focus is the approach that we take to investigating these genes, the technology that we use, and the way that we translate that information. We also, in terms of our research, go all the way through from population, where we actually get samples from people who are affected with disorders, through to functional studies, where we look at what's going wrong with genes. We obviously have a clinical component because we need to work with clinicians to get hold of information and samples that will help us in the studies. But we also do something called pharmacogenomics, which is to try and tie the information that we get from genetics to potential treatments. So it's actually under a banner of what people would now call personalised medicine. The sorts of disorders that I mentioned are called common complex disorders, and that's not just because they're tricky. In genetic terms, it means that they're multifactorial disorders that involve usually multiple genes as well as environmental factors. So there may be triggers or influences from the environment. As an example, if you look at something like high blood pressure, affects about 20% of the population, has a strong genetic component, probably about 60–70%, but there are clearly environmental factors associated with high blood pressure. It's related to things like your diet, your salt levels, your exercise levels, whether you smoke, as well as your family history. So there are environmental factors associated with these disorders as well as multiple genes. In order to identify the genes involved in these sorts of disorders, it's really important to have well-characterised and large populations to support these studies. And so far we've collected over about 10,000 DNA samples from case control populations, also from large families and also from specific cohorts. So as an example, for our breast cancer studies, we're currently collecting every prospective breast cancer case in Queensland over about a 3.5-year period. So this is a study that we're doing in collaboration with Cancer Council of Queensland. Every person who's diagnosed with breast cancer is invited to be part of the study. We obtain medical, clinical information, family, lifestyle information, but we also contain blood samples for DNA studies, for gene expression studies, for epigenetic studies, etc. So we're establishing quite a large prospective biobank for breast cancer. We have similar studies that we're doing in MS, where we use brain tissue samples, where we look at gene expression differences. We've also been involved in a study using about 5,000 people from the town of Nambour, and these have been followed over a longitudinal period for about 5 years, where we looked at the development of skin cancer in these people and related it to lifestyle factors and to family history, but again to genetic factors. One of the studies that we're probably most well known for is our studies on migraine. We probably have the largest migraine biobank in the world at the moment. We have over 100 large families. We currently routinely use 1,200 matched case and control populations. We've got over 3,000 cases in our biobank, and a lot of information from these people because they're very keen to be involved in research to try and identify new treatments. We also have samples from an island population from Norfolk, and I'm going to come back to that one in a minute. So all of these studies are what's called gene mapping studies, and what we're trying to do here is identify genes by using these populations. So it's important that the populations are pretty well characterised, and that's actually a key to these sorts of studies. So I really love very large families. I'm a geneticist. I get really excited when I see multiple generations and multiple affected people, because these sorts of populations are very powerful to help you try and track what's going wrong with the genes from generation to generation. So as an example, my husband's last name is Smith, so as you'd expect, he's got tons of relatives, okay? And so we went to a family reunion a few years ago, and he has 8,000 living relatives. That's pretty exciting, okay? So I immediately started getting their DNA samples, pulled out some syringes from the back of the car, set up a little stall, and I've been to several of those reunions now, and that's one of our families that we actually use for some of these studies. But another of families that we actually use from our studies derive from a very famous historical event, and that's the Mutiny on the Bounty. And for those of you who know your history, and if not, you can probably see it through various films, because there's been a lot of really good films about this. And there's ones with that depict Mel Gibson in there, and there's other ones with Marlon Brando, etc. But what happened was that in the 1780s there was a ship called the Bounty that was travelling originally from England going across to the West Indies, stopped in the South Pacific around Tahiti, and then they had a mutiny. So Fletcher Christian led the mutiny, and some of his friends decided that they really didn't like the captain, who was Captain Bligh, that he wasn't treating them well, that they quite liked all the people that they'd met in Tahiti, and that they'd get rid of him, take the boat, as well as some nice Tahitian people, and take them across to a very small island called Pitcairn Island. So Pitcairn Island is located between New Zealand and South America. Sort of middle of nowhere. They sunk the boat just in case anybody saw them, that there weren't too many people around to see them. And then they proceeded to have children and more children, and a few more wars, and then a few more children, etc. And in Queen Victoria's time, they were rediscovered. And by then, Pitcairn Island was getting really full. There were a lot of people there, there wasn't much food, and there wasn't much water, and they were really sort of in dire straits. So she pardoned the ancestors and gave them an island, called Norfolk Island. And Norfolk's located about 2 hours east of Brisbane. It's a very small island. It used to be a penal colony for New South Wales for very, very bad criminals, but it got a bit messy and bloody, and it was also very hard to get information to them because it doesn't have a natural harbour, so you have to actually have big ships get stuff on them, and then you put them on little ships, and then the little ships are rowed across to the island. So it's very hard to get food and things there. So it was actually an island just sitting there. Nobody was on it. It's very pretty. It's in a very nice spot, and it didn't belong to anybody except that it's a territory of Australia. So Queen Victoria gave them Norfolk Island. Majority of people from Pitcairn moved there, and they're still there, and their ancestors are still there. So basically what we have is a geographically remote, isolated population with known founder effect. By this we know who started that population. And in fact there's 21 founders. There are 9 paternal or male bounty mutineers and 12 Tahitian women. So those 21 founders gave rise to the current population of Norfolk Island. And in fact 80% of the people on the island can trace their ancestry now directly back to the original founders. If you look at the telephone book, you can see that there are very strong family groupings, and they also have a very well documented family history. They're very proud of their genealogy. But if you look at the telephone book, there's about 5 last names. Okay, so it's very strong family groupings. And in fact, the majority of people, about 80% of them, fit within a single 12-generational pedigree that has about 6,500 people in it. It goes back to the original founders. So for me, this is a really exciting sort of family. I met somebody at the pub who came from there, as you do, got interested in it, spent a couple of years talking to the people on the island, and we ended up in the year 2000 for my very first sabbatical as a researcher, setting up a clinic on the island. So what we did in that clinic was take over absolutely everything, the very first computer on the island, centrifuges, freezers and everything. We provided a medical health check for every person on the island because they didn't have that. Nobody had checked their blood pressure, nobody had checked them for diabetes levels. Sorry, Martin. I had to start, they made me. So basically we set up a clinic on the island because at the time Norfolk had a single doctor on the island, so it's emergency medicine only. So they didn't have the normal public health screening that we normally have. So what we decided to do was to actually provide this information back to each individual, give them a full medical check. We did this at no cost because they were helping our studies. They were helping us to get DNA samples for our genetic studies. And so that's what we did. And we set up a family structure. Now this is actually not the normal way to draw pedigree structures. Normally you have circles and squares, and the circles are the females and the squares are the males, but it's pretty complex when you try and fit about 6,500 people in 12 generations. And so if we were to draw that out, it would actually be about 42, 43 pages around this room, and it would be too big for me to show you. So this is our shorthand way of showing that it's a very complex pedigree. And in fact, when we look at that pedigree, we also can look at, well, who has disorders in this pedigree. And I mentioned that we have an interest in migraine. Now, normally if you look at migraine in a Caucasian population like Australia or New Zealand or America or Europe, you'd find about 12% of the population is affected, which is a pretty common disorder. But if you look at an isolated population, where everybody, well, the majority of people fit within this single pedigree, then there's limited extra genes coming in. And so what you find, and why I get so excited by a population like this, is that if you have genes for disease, they will tend to be amplified. And this is exactly, here's an example. Instead of being 12% of the population affected by migraine, you have 25.5% of the population affected. So this is an amplification because it means that there are genes there that are being kept in that population and being amplified going through the generations. And this is why geneticists like me get excited by what's called founder effect isolated populations. So you may have heard quite a number of years ago, there was a company set up called Decode. Anybody ever heard of Decode? A few other people have as well. So Decode is a company that was set up, and it just capitalised on the fact that Iceland is an isolated population. So they actually made a company out of the whole country. They had information from all the people in Iceland. People were very happy to give permission to actually use DNA for various studies. They also have really good clinical and medical information on that particular population. So they actually turned it into a commercial company and called it Decode, and have used the Icelandic population for many genetic studies. In actual fact though, Decode's probably got too many founders. The best isolated population is believed to have between 20 and 25 founders, and Norfolk has 21, so we actually think it's a better sort of population. It's more likely to have amplified genes, and that's why we've been using it for studies like migraine and also cardiovascular disease. And just to show you sort of what we've done with that, we've recently done a genome-wide association scan in the Norfolk Island population. And what we've done there is look at something like 700,000 SNPs across all of the chromosomes. And we've recently identified a new gene, a novel gene, adenosine deaminase gene, that actually edits or modifies two neurotransmitter levels in the brain. So one is the serotonin receptors and the other are the glutamate receptors. So this was identified by just using that population. It's a gene that showed significance across this whole thing. We've published on this, and we've actually shown that it actually has a candidate gene or an effect in the neurons in brains, although we haven't got to brains yet, but we've shown that it has a potential effect in this area. So the sorts of studies you can do with this large family is to look at whole genome things like this in terms of genome-wide association studies. And here's the result of that one, where we looked at markers across every single chromosome. The little dots up the top are the exciting bits, and one of those related to the gene that I just mentioned, the adenosine deaminase one. But it wasn't just one marker, it was a whole block of markers within the promoter of that gene showed association. We also can do other studies with Norfolk populations. So we've looked at cardiovascular disease risk traits, and we have about 36 traits that relate to cardiovascular disease. And we currently have a couple of papers that are under review in relation to new genes that relate to them. We also are currently doing not just GWAS studies, but are doing sequencing studies. So we're currently just about to start whole genome sequencing for migraine and also for glaucoma. So in migraine we have a region implicated, a particular gene I showed you, but also a region on the X chromosome. In glaucoma we're going to go across the whole genome to look for new genes that relate to ocular phenotypes. We've also commenced epigenetic studies, And we've also commenced whole mitochondrial studies too, to see if there are mutations within the mitochondria related to disease. We also don't just look at genomics, we also look at expression. So we do something called gene expression analysis, and it's called EQTL analysis. So in terms of why we would use this population, we use it because it's a very powerful population to try and track down genes involved in disorders. And we then use our other populations to see if what we found on Norfolk applies to more common outbred populations. So what I'm going to focus on just towards the end here is I mentioned migraine, and this is probably the area where we're known for most. As I mentioned, it's a really common disorder. Overall affects about 12% of the population, but probably the largest study has shown about 18% of women are affected and 6% of men. That's a really significant number of people affected by this disorder. And there are no simple laboratory-based diagnostics for migraine. It's actually, you define someone as having migraine by what they tell their doctor, or getting rid of anything else that it might be. There's also some very good treatments, but they only work well for about 30 to maybe 50% of the population. So there's clearly a need to develop new treatments. There's two main types. As you'd expect, there's head pain, and it can be very severe and go right up to about 3 days in duration. It's also one side of the head, unilateral and throbbing. It's also associated with nausea and vomiting and photophobia and phonophobia. And in addition to that, people who suffer from the least common, it's about 30% of them, migraine with aura, also have a number of weird neurological disturbances. So just before the head pain, they'll tend to get visual disorders, so flashing lights, wavy lines, coloured sights, tunnel vision, even complete loss of vision is part of this. They also have trouble speaking normally at the early stages of a migraine. And they can have weakness through parts of their body, numbness, and even paralysis, usually one-sided on the body. This is actually all part of migraine. But you can see that some of those symptoms are starting to look like other more severe disorders. It starts to look like potentially brain-type tumour disorders or potentially a stroke-type disorder. So sometimes it's hard to define what you're actually looking at, particularly if you're looking at a young child who's got paralysis of part of their body, who can't speak properly and who've lost their sight. You know, you're having some troubles there. So we were interested in identifying the genes. At first we were hoping it'd be a single gene, it would all be really easy, but we've been at this for 15 years and we now know it's not so easy, that there's actually multiple genes, that it is a complex disorder. It does have environmental factors, so things like certain types of food can be involved in triggering a migraine in those who are predisposed. Usually the good foods like red wines or chocolate or nice cheeses, all the good bits, which is pretty sad. But also it can be certain types of smells. Sometimes strong perfume or cigarette smoke can actually be the trigger to bring on a migraine in people who have the genetic background. It can even be things like barometric pressure changes, major changes in the weather, etc. The disorder shows comorbidity with things like epilepsy, also with cardiovascular disease and in particular stroke, and also with depression. So it could be that there are shared genetic factors here. In terms of the genes, we don't know the number of genes yet, but there have been a number that have been identified, and we've been involved in some of those. And to do this, this is a traditional picture of a family. To do this, we use families like this, where the people coloured in who are other people who are affected. You can see that it tends to occur in every generation, although occasionally it skips it. So it could be there are interacting genes that play a role in actually people expressing those particular genes. Using those sorts of families and also using the Norfolk family, we and others have implicated genes across various chromosomes. So one of them recently, a genome-wide association study, implicated a glutamate receptor on chromosome 8. I showed you that we implicated another one that also relates to glutamate receptors. And we were also involved in a study that implicated a potassium channel gene that has mutations that are clearly inherited from one generation to the next. Anybody who has the mutation has the disorder. So from that, at the moment, we currently have 3 known, and this is FHM is familial hemiplegic migraine. It's a subtype of migraine with aura. It's very severe. It's usually manifests in young children, 4 or 5 years of age originally. They have very severe migraine. They have I mentioned sort of paralysis. They usually have paralysis across half of the body, but they also have severe problems with their senses, so that sometimes they can lose their vision, they can lose their hearing, and it actually can be extended across for months. They can also go into coma for months as well. This is all part of migraine, and it's all due to specific genes. So at the moment there are 3 known genes for familial hemiplegic migraine, and they all relate to genes that are expressed in the brain, so channel genes. And so these are, one's a calcium channel gene, one's an ATPase gene, and the third one's a sodium channel gene. That third one I mentioned is particularly interesting because mutations in that cause this very severe type of migraine. They also cause epilepsy as well. And that could explain, I mentioned that there's comorbidity between the two. Well, it's the same gene that can cause both of them. The first gene I mentioned, that the calcium channel gene, also causes not just this type of migraine, but it can cause some other ataxia, episodic ataxia type neurological disorders as well. So it's important to look at these genes to diagnose people, and there are treatments directed towards these, so to then define for a clinician what sort of treatment. So we currently do diagnostic testing for all of these genes, and we do it for Australia and New Zealand and parts of Asia as well. Up until recently, we've done this by using the normal technology or gold standard technology, which is Sanger sequencing, and we would test for known mutation points. This is NATA accredited facility, and it's also Human Genetics Society listed. So all the samples in Australia come to us. They come through pathology labs all around Australia, or the clinicians send the samples directly to us. We then analyse those and send the results back to them. So that's one area in terms of genes, and that's some diagnostics we've been able to translate. Other genes that we've replicated, probably 3 classes. They fit within the neurotransmitter class. But we also, a few years ago, looked at hormone receptor genes. If you think about this, there's 3 times more females affected than males. And so when you look at that, you'll find that females also have a significant hormonal influence, because the majority of females who start to develop migraine do so around about puberty. And in fact, women who suffer from migraine find that it actually changes when their hormonal levels fluctuate. So there's actually something called menstrual migraine. It occurs in about 20% of females. There's also when people go on the pill, when they go off the pill, when they go on HRT, when they get pregnant, their migraine levels will tend to change. So there's a hormonal influence. And we showed specific variants in both the estrogen and the progesterone receptor genes play a role in migraine development. And we believe in particular that relates to the menstrual migraine category. We're currently doing a very big trial with a collaborator in London, in the London Migraine Clinic, collecting samples, and we're just at the moment actually analysing those in terms of these genes and expression of those genes. But the very last one I'll show you, and that has had some commercial significance as well, is probably the 3rd class of genes, and that's the vascular related genes. So migraine people, particularly migraine with aura, show blood flow differences in the brain. They also show something called cortical spreading depression, and it's a wave of depolarization that goes across the brain just before the migraine actually starts. So we thought, well, maybe we should be looking at genes that relate to blood flow differences. And one in particular is a gene with a very long name, methylene tetrahydrofolate reductase. It's a gene that's been shown previously to have this very significant functional mutation. If you have this mutation, the C677T, it results in about a 50% drop in your enzyme levels, which is the product of the gene. Those low levels of enzyme affect folate levels, increase homocysteine levels, and that's been associated with increased risk of stroke and heart attack. And we decided to look at it to see if it had any effect with migraine as well. Did it play a role? Because some of the symptoms you see between the 2 disorders are similar. So when we did this, there'd been a couple of very small studies, one in a Japanese population that showed that mutation occurred much more often in those with migraine. Similarly in a Turkish population, and we then extended it out by looking at a larger Australian case control population, but also 82 families as well. And in all of this, we showed a significant association of that mutation with migraine. Not with migraine in general, but only migraine with aura, and that's the one that's associated with stroke as well. So it made a lot of sense. And subsequently, we're not the only one. It's now been done in many different labs around the world. And in fact, this is the only gene that stood up as a susceptibility gene for migraine. And this has been done in over 3,000 migraineurs in many controls as well. Okay, so that's pretty interesting. Here we have another gene and it plays a role in migraine. But more importantly, this particular gene was studied in about the 70s and 80s in a lot of biochemical studies. And it was shown in detail what pathway it fits in and how it works. And more importantly, the cofactors were identified for this particular enzyme. And the cofactors are actually vitamins. B6 and B12 are cofactors for this enzyme to actually work. Also, it fits in a pathway that involves folate, and folate actually acts by a feedback loop to actually affect the enzyme activity as well. We didn't do this, other people have done this. But what we did was we took it one step further and we thought, look, anybody who has this mutation has high levels of homocysteine, and this puts them at an increased risk of stroke. So if we add some more of these cofactors, can we make the homocysteine levels go back to normal and reduce the risk of stroke? And if we did this, would it actually have any impact on their migraine? So we did a double-blind placebo-controlled trial over 6 months where we took people with and without this mutation and suffered from migraine with aura. We gave them folate, B6 and B12, and when you give them that over 6 months, oh, what do you know? You increase their folate and their B12, so it's just showing. And their B6. But more importantly, it significantly dropped their homocysteine levels. So we found that it happened in the placebo group, and probably because they were thinking about this and eating more B group things, but it wasn't significant in the placebo group, but it was in the treatment group. So it had a really good impact on their actual homocysteine levels and made it normal. But more than that, it had a really good impact on their migraine. It dropped the migraine frequency and the severity and the pain associated with it significantly, and only in the treatment group and not in the placebo group. That was a trial of it was 52 migraine with aura patients. It was very significant and it was really quite promising. So we went to the next step. We went to a phase 2 trial then. So we then took a trial involving about 250 patients. Again, migraine with aura. We focused on females in case there was a gender difference, and we kept them stuck with Caucasians in case there was an ethnicity difference. And we followed again 6 months, same sort of treatment, placebo-controlled over a 6-month period, showed exactly the same thing. Significant decrease in migraine pain, severity and frequency as captured by a particular way of measuring this, which is called migraine disability. And you could look at those individually, severity of pain, etc., or you could look at those together in the disability score. But the interesting thing was exactly the same thing happened. Again, we were able to show that the homocysteine levels dropped, but because we had a bigger population, we could start to tease this out a little bit more. So everybody has 2 copies of every gene, and so people could have a single copy of this mutation, or you could have a double copy if you get it from both parents. So what we found was that people with the single copy actually responded better than people with the double copy. So to us, this made us think, well, maybe we need different doses. So if you've got a double copy, maybe you need a higher dose than people who have a single copy. And so looking again at teasing that apart, we showed this in terms of genotype as well. But the most important thing that came out of that is that we're now currently doing a phase 3 trial, which is a dose-dependent trial. So we currently have 600 people involved in this trial, so it's a 6-month double-blind placebo-controlled trial. We have a high dose, we have a low dose, and we have a placebo. And we're really hoping at the end of this that we'll be able to look at these results and actually have a product on the market there. We finished the last person, we screened over 1,000 people to be involved in this next trial. We've got them all involved in the study, and there's actually 600 of those in the 3 groups. 3 groups I mentioned is high dose and low dose. And we've gone all the way through from recruitment to baseline collections to actually doing the trial to following them up over a 3-month period and to getting all the information at the end. We're currently about to break the code on that because the last person finished in April, and we should be able to analyse those results soon. All of this, though, was done with a partner. And the partner, because it's a nutraceutical type treatment, was Blackmores. And Blackmores came in right from the beginning. I approached Blackmores because I'd heard that they were doing a study using this sort of treatment to look at the potential on stroke, and we were interested in looking at it on migraine. So they've been our partner through the phase 1, the phase 2 and the current phase 3. To get it funded originally was pretty tricky because it's a small trial that is really kind of a new pharmaceutical, and so it was hard to get through NHMRC, but we had the Brain Foundation support us, and even Janssen's, who is a pharmaceutical company, supported us for the first trial. Second trial we got through the Nutricia Foundation, and the third trial is actually being funded as part of Therapeutic Innovation Australia, which is translating health discoveries through ARC, but also with Smart State funding. I know Smart State's gone away now, but there's still a little bit of Smart State left, and this is probably one of their last projects. Although I think it could still be very smart. So what we've got here is a potentially promising treatment, and obviously it has potentially commercial potentials here. So we've got a migraine therapy. We also have a DNA diagnostic test, because I mentioned that this is due to a gene. This only works for people who've got the gene mutation. So as part of our panel of genes, we do a whole range of genes for sequencing, but we have a simple single test set up for this one as well for us to detect it. And again, that's a NATA-accredited DNA diagnostic that we believe will sit with this personalised migraine treatment. The proof of principle is that we can look at nutraceuticals and pharmaceutical therapies tailored to treatments for migraine. And use the information from DNA to actually define this. So DNA diagnostics going hand in hand with actual treatment. So we think that this commercialisation pathway is really for a targeted treatment with a DNA diagnostic. I mentioned that we do diagnostics for the other disorders, so FHM 1, 2, 3. We also do it for episodic ataxia type 2, spinocerebellar ataxia type 6, and CADASIL. All of these show symptomatic overlap with severe migraine, and that's why we do all of these together. Now, we used to do each gene individually. It was time consuming and very expensive. It would cost us $2,900 to just do the 6 genes that we currently do and just look at the known mutation points. We've recently switched over to using next generation sequencing and set up a panel that allows us to do all those genes in one hit, does it much faster, has much better coverage of all of the genes, and it currently costs about $700, and you can get the whole lot done in one. So instead of a clinician saying, I want you to test for familial hemiplegic migraine 1, we would look at these exons, we would then send them back a bill and say, sorry, that was negative. Then they'd say, can you look at your extended coverage? So we'd go to the next set of exons, then we'd go to the next, and then if that was negative, we'd go to the next gene. So it was a long, iterative process. So now what we do is the whole thing in one hit, and it's much cheaper. And it's a really, I think, a very good way to do it. And as I mentioned, we also do the MTHFR testing there. And probably just the last thing I want to get across is, commercially, we do diagnostics, but we clearly have other capabilities in our centre as well. So we do things like genotyping, sequencing, expression studies, epigenetics. We can do it whole genome or individual genome. We can do mitochondrial. We obviously have expertise in extractions and biobanking, but we have a whole bunch of equipment because we're part of something called Therapeutic Innovation Australia, and we got ARCE funding to set up a service facility so that other people can access it. And so all of these things can be provided as a service to people who want to use them. And all that equipment has moved up here to QUT and is available for that. We also do our own biostats and bioinformatics, molecular diagnostics and clinical trials I mentioned. We do clinical trials for all sorts of things as well. So not just for migraine, it's not just research investigator driven. We're about to start a large be a centre that's part of a large multinational Alzheimer's prevention disease trial. And this is a prevention that's based on, again, a particular gene problem. It's going to be a 5-year trial. We'll be following hundreds of people for 5 years looking at whether this particular treatment can actually stop development of Alzheimer's. We've even done studies sponsored by coffee companies to look at the effect of caffeine on gene expression, etc. So once you have some capabilities and expertise, you can link with industry partners to do all sorts of things. I've listed some of our industry partners. Some of them have been very big. GlaxoSmithKline, Janssen's another one, Merck. Some of them have been smaller companies that we work with. The Amarillan one was the one that we worked with to identify the TRES potassium channel gene involved in migraine, etc. We've even worked with Corbett Research as a beta testing centre for their equipment for quite a number of years. So as I mentioned, all of this fits in something called Therapeutic Innovation Australia, and all of it's at QUT, available to QUT people, but also external. So I mentioned they have a team. I work with clinicians. Top 2 are all clinicians, but also a fantastic team within my lab, and also my bioinformatics people and people involved in collecting things, as well as funding sources. So thank you very much.